I'm very happy to welcome Kritika Garg. Kritika was a PhD student here at NCBS where I am a while ago, and she's now been a postdoc in different parts of the world and has started her own lab at Ashoka University just last year. Kritika works on understanding the distribution of different animals, primarily bats and birds so far. So looking at animal distribution from a biogeographic perspective, and she uses usually a lot of molecular data. And in the past, she's also done some behavioral work to understand how animals distribute themselves and what factors determine where they're found and how many of them are found. And so she's going to tell us about the impact of Pleistocene cycles, glacial cycles on bird species in Southeast Asia today. So welcome, Kritika, and everybody please feel free to unmute yourselves and ask questions as you go along.
Hi, thank you for the introduction, Deepa. Good afternoon, everyone. I hope I'm audible. I have a sore throat, sorry. You'll have to bear with me on this. So as Deepa said, please feel free to ask me questions whenever you want. We can go slow, fast, depending on how you follow, all right? So good afternoon, everyone, once again. So today I'll be talking about Pleistocene glacial cycles. Now, these are cyclic cooling and warming periods that we see for the past 2.6 million years. So the Pleistocene roughly period corresponds from past 2.6 million years to last 10,000 years. After that, we have the Holocene, 10 to 11,000 years back. It's termed as Holocene. Now, during these cooling cycles, what we have is the water gets locked up as ice, and most of it is locked up in these polar caps here. So you have water being locked up here in the northern in the Arctic Circle as well as in the Antarctica. And a lot of these higher mountain range also gets glaciated. Now, because water gets locked up in ice over here, the sea level actually drops. So your current sea level, we are right now in a warm phase, that's the interglacial. So the current sea level is at 0 meters. But whenever you have this cooling cycle, the water drops by up to 120 to 150 meters. So a lot of these islands, especially in Southeast Asia, get connected. So this is what would be the landmass that would be visible whenever you have a glacial cycle. That's the cooling period. So 20,000 years back, we had the last ice age. And at that point of time, all of this landmass was available. You had forests growing here and slowly. You had animals that could move around. And one of the best ways to know that, the glacial cycles actually occurred and you had this land bridges which are used is actually you see orangutans with a very disjunct distribution, or even your rhinos. So you find them in Sumatra, then you find them in Borneo. So they're able to use these land bridges that are formed and move across. Now, these cycles have played a really important role for your current biodiversity. A lot of species are really young. Some of these species have evolved around 2 million years back. Now, in this graph that I'm showing you, these are your white ice. You're looking at these Soptrus genus. This is found throughout the old world tropics. And there are about 150 species as of now. So they go to one island and then they become a new species. So these are called the mice in your bird literature. So these are your mice of birds. So they go to one island, they hop onto a new island, then they become a new species. Then they hop onto another island, become a new species. So all over in Southeast Asia, you have multiple different species. And what basically you see in this plot here, this is a lineage by time plot. Basically, you have the time here. So this is 10 million years to current, and the number of lineages being plotted. And suddenly around 2 million years back, you have a lot of species that come up. So these cooling and warming cycles have led to species getting isolated, populations becoming isolated, differentiating, extinction also taking place during this period. So over here, this is how hopefully Earth looked like 2 million years back. So this is just a painting by someone who has shown what Earth would be like. So these cycles have been very important, and a lot of work has been done to try and understand how they shape the current biodiversity. But the problem is most of the work is from the Northern Hemisphere. A lot of our understanding comes from Europe, and some bit of it comes from America. But a lot of this,
Hi.
Yes.
I have one stupid question. So
please go ahead.
I mean, irrespective of whether it's a warming cycle or a cooling cycle, birds can fly, right? So how would it restrict their movement? That's my question.
All right. So that's a very good question. Now, not everyone can fly. the distance. If you look at the birds in your background itself, in your backyard, not all of them can fly long distances. So those who can fly long distances, maybe they get shielded by this, but not all species. And there are about 10,000 species of birds, all right? A lot of them are within your tropics. So not every species can actually withstand these cooling and warming cycles. So their habitat actually shrinks or expands depending on which part of the world they are located in. So that's something that we'll get into later on. I'll show you like how, different populations in the same region actually respond to these glacial cycles. Not all of them show the same response. It depends on the species biology, their requirement, things like that. All of that come into play. Is that okay?
Yeah,
all right,
sure.
We'll look at those. So now, as I was saying, a lot of our understanding from the impact of these glacial cycles actually come from the northern hemisphere, where you had these large ice sheets that were formed during the glacial period, and most of the species had to retreat to these small refugia here. Basically, the refugias were here, and much about what the impact of these glacial cycles in the tropics is not known. And tropics are where most of your species diversity is. So in the past 10, 20 years, there has been a momentum to try and understand the impact of these glacial cycles in the tropics. So I'll be discussing a few studies that I was involved in, leading some of them in Southeast Asia. All right, so we are going to look at the impact of these glacial cycles on islands in Southeast Asia. So the first group of islands that we are looking at is next to Sulawesi. So Sulawesi is here. So this is just for your geography. Your Borneo, Sumatra, Peninsula Malaysia, your Borneo, your Java here, and you have this very funky looking island, that's the Sulawesi Island. And next to it, we are looking at these two small islands. Okay, so these are part of the Sulawesi chain, Sulawesi Island. So they are called as Peleng. So this is the Peleng group of islands, and this is the Taliabu. So these group of islands, basically every time the sea level drops, so whenever you have the cooling period, the sea level drops and they get connected. Otherwise, they get disconnected. Whenever you have, so at the current warming cycle, they get disconnected. So we are looking at two birds here. One is a forest specialist. The other is can tolerate edge habitat. So the forest specialist is called the henna-tailed jungle flycatcher, and the other one is called the golden whistler. Now, what is interesting about the henna-tailed flycatcher is that it's only found on these two islands. It's not found anywhere else. The golden whistler is found all around these small islands as well as in Australia. But we are looking at subspecies of this which are found only on these islands. Now, we want to actually understand how species biology may interact with these glacial cycles, and how does it affect the population structure that you see in these species. So just remember, your henna-tailed flycatcher, jungle flycatcher, likes good quality habitat. So it requires a good quality forest cover to move around, whereas this species is tolerant to poor quality habitat also. Now, how do we go about investigating this? So for this, we are looking at two different markers. One is your mitochondrial marker. So we are using the barcoding gene. So in birds, the barcoding gene is generally CO1, COX-1 gene. And we've got the sequences for this using amplicon sequencing. So this was a part of another big major project where we're sequencing all the samples in the lab. So we just did an amplicon sec because it was very cheap to do it compared to Sanger sequencing. And then for the other one, we are looking at something called a single nucleotide. Sorry, I need to minimize this. Yeah, all right. All right. So is the screen visible now?
Actually, no. Now I'm seeing just a black screen, and then I think your slide is in the background somewhere because I can see something on your slide, but not.
All right, let me stop sharing and sharing again. Stop sharing. Is this better now?
Yes, clear.
All right. Yeah, sorry, I had to minimize the panel, otherwise I can't see half of my slide. All right. So here we are looking at two different markers, as I said. One is the mitochondria. So the unique part about mitochondria is that you will only get the history of all the females because it's inherited from your mother. So we are looking at that history, as well as along with this, we're going to look at the nuclear genome history as well. And for this, we are looking at single nucleotide polymorphisms. So just as a recap, I hope you have learned about this now. So basically, you're looking at anything, you're looking at tracks of sequences across individuals, and you're interested in markers which have one nucleotide, they vary at one nucleotide. So in this case, you have this polymorphism where you have a G and an A. And generally for all the work that I'm talking about today, we are looking at biallelic markers. So we are not looking at triallelic or the variable sites which have all four possible nucleotides. We're only looking at biallelic markers. Now, how do I get this? So we use a method called as reduced representation libraries, where we use a restriction enzyme to digest your DNA across all samples and only sequence a small portion of your genome associated with this restriction site. If you guys want to know more about these methods, we can always go into details at a later stage. Just to let you know, this is one way of getting a small fraction of your genome. You can target anywhere with 0.1 to 10% of your genome this way. So you don't have to sequence the whole genome. You can sequence a small portion of your genome and get these polymorphic sites. Now, looking at the mitochondrial DNA, so we sequence 313 base pairs of mitochondrial CO1 gene. And what we see for this is the forest specialist. And you see that the haplotypes actually are very distinct. So the island on Pelling compared to the birds when we captured them on Taliabu, they have a very different mitochondrial CO1 gene. And the number of differences, if here you quantify them, is equivalent to saying that the population present on Pelling is a different species compared to the one present on Taliabu. Whereas for the forest, the H species, which can tolerate poor quality habitat, the differences are few. You see 1, 2, 3, 4, 5. So you see 5 differences that you can see between the two closest haplotypes. So basically, there's not much difference between the two island populations here. And if you get into the nuclear genome, so here I'm looking at structure plots. So what we are looking at is each bar is one individual. And over here, we look at this proportion, that's the ancestry proportion. So depending on the colors, we can decide which ancestry each individual has. If it has a mixed color like this, 2 colors, then it has a mixed ancestry. Whereas over here, this is just one single color or one single color here. That means this individual has only one type of ancestry. So if you look at this forest specialist, again, what you see is the birds from Pelling have one kind of ancestry. They are all orange. Whereas from Taliabu, they're all blue. They have a different ancestry. So basically, even in the nuclear genome, they're highly differentiated. Whereas for the forest H species, you don't see a lot of differentiation. You do see that, Pelling are more orange compared to blue, whereas Taliabu is equally, both colors are equally likely presented here. But generally, there's some sort of differentiation, but it's not very high, not like this. This is clear cut. Pelling is very different from Taliabu. And basically, you can take these markers and actually do a modeling. So here we did a model for gene flow and isolation. So what we did is these rat tags that we have a short sequence that we get. We get about 120, 130 base pair sequence. And we have about thousands. So there was about 3,000 markers for this species and about 2,000 for this. And we can use them to model and I'd understand when the divergence of the two populations or the two species took place and what's the rate of gene flow. Now here you're assuming continuous gene flow. All right, so it's a very simplified model. And what we see is the divergence for the forest species is much older compared to your forest H species. And the level of gene flow is very low. So there's hardly any gene flow in this species if you see the henna-tail flycatcher, jungle flycatcher. Whereas over here in this golden whistler, you see a lot of gene flow from Pelling to Taliabu. It's a unidirectional gene flow, but there's a lot of it. It's about 44% of the population from here moves into there, actively reproduces here. So there's a lot of movement of individuals. Now, the other thing that we actually saw was when we look at the timing of isolation, the timing of isolation actually corresponds to the stages where the water level was high. So if you go back to this, whenever you have interglacial, the warm period, these two islands get disconnected. So there is a water path here. So there's no way the birds can move. They don't fly that much, all right? They can only move when there's a land bridge. So what we are seeing is, the divergence took place whenever the sea level was high. So those are the important things that we can actually infer using molecular data here. So what we see is the ecological requirements of the species drives the level of isolation. The species which requires good quality habitat, so requires the habitat to be of pristine type, then it cannot move across. So once your land bridge is formed, it's just an open seabed, right? So if the species just cannot just move on that seabed, so you have to wait for the forest to grow. What kind of forest is formed? So first you'll have your grasslands, then it will then you will have your deciduous species, and then your primary forest. So it takes time. So a species which has a higher quality requirement of forest cannot move that easily, and so it will remain isolated even if the land bridges are formed. So that's what we see. The forest specialist, you have a high level of divergence, and when you look at the level of divergence, it's a species level, whereas the edge species can tolerate poor quality habitat and has a low divergence. Now, the divergence also occurred when the two islands were isolated. And these land bridges are semi-permeable. That means if the land bridge is formed, it doesn't mean automatically everything can move across. Only species that can use the land bridges can form small populations, established populations in these land bridges can move across. So this is one thing that we noticed. Now, the other thing that I'm getting into, so sorry, I'm going to jump into our next study. Before I get into that, is there any questions here?
Kritika, I have a general question.
Sure.
Is there a difference between the body size of these two species? Is that something that plays a role?
Actually, we did look into this. So the two species, the one we selected such that, they are of similar body size and their flight capabilities are also similar.
Okay, thanks.
All right. So I'll get into the next study. So the next study, we are going to look at this region. So we were looking at Sulawesi here, the satellite islands of Sulawesi here. Now we are getting into something called as the Sundaland, which consists of your islands of Sumatra, Java, Borneo, and part of Peninsular Malaysia, and also this island from Philippines, yeah, Palawan. So this is your Sundaland. Now this, whenever your sea level drops, you have this whole land that emerges. You have a lot of these river systems that paleo channels that are active. Now, what is interesting about this area is that, if you look at just this landmass, if you look at these islands, whenever you have the sea level dropping, this actually doubles in size. So you have a huge area of land that becomes available for species to occupy. And we are going to look for this study. We are looking at two babblers. One is a short-tailed babbler, one is a black-capped babbler. Now, both of them are forest-dependent species, insectivores. They prefer the understory in a forest habitat, and they have very similar ecological requirements. And what we are interested in is, now in these between these major islands, right? There are few small island complexes here. One is the Natuna Islands, the other is the Anambas. Now, these islands currently, these species are found, but we don't know from where, which populations have come and colonized. Are these closer to Peninsular Malaysia, or are they closer to Borneo? What their affinity is, or are they different species altogether now? They diverged so much from the rest of the populations, they're altogether different. We don't know anything about these small islands here. And the other thing that to notice is the Natuna is closest to Borneo. Like if you look at the distance as a crow flies, it's about 220 meters or something, kilometers or something like that. Whereas compared to Peninsular Malaysia, it's almost double the distance. However, if you look at this sea level drops, different sea level drops, Natuna Islands and the Anambas connect to Peninsular Malaysia earlier. So there is a small channel here, marine channel, which actually prevents the connection immediately to Borneo. It takes about 1,000 odd years further, or a sea drop level of around another 10 to 20 meters at least, for these two islands, for these islands to connect to Borneo. So generally these islands, though they are, yeah, Deepa, please go ahead.
Sorry, I don't have a question, but there's a lot of background noise that I think your mic is picking up. Is it possible to close the door or something if it's open?
I'll just try. 1 second, I'll try. Sorry, I hope this is better now.
Thank you. Thank you.
Yeah, there are a lot of kids playing around and they won't leave, so you can't do much. All right. So what I was saying is these Natuna Islands, these small islands, Natuna Numbers, they're quite close to Borneo, actually the island of Borneo. But they connect to Peninsula of Asia first when the sea level drops. So that is something that is important to remember. All right. Now we are looking at two different species of birds which have very similar ecological requirements, unlike the previous study. Now, to do this, actually, it's very difficult for us now to go to these islands, these small islands, and get samples because most of these islands are converted into these really expensive, fancy resorts. A lot of islands in Southeast Asia are converted like that. So you don't get permits to go sample there. Then there's a lot of logging that happens as well. So, it's difficult to capture these birds now. So it's easier to actually go back to museum samples, 100-year-old, 200-year-old museum samples, where a lot of these areas were collected. You had people moving around from this area. You had Wallace doing a lot of collections, actually. His collections are also found in the museum at Singapore, the Lee Kuan Yew Natural History Museum. So what we did is we actually used museum samples for this work. Now, what is the problem that we face now is as soon as we get into ancient DNA, especially for non-model organisms, is the DNA is of very poor quality. So this is what the DNA profile looks like when I extracted one of these samples. So this is what the profile is. So most of the fragments are around 100 base pairs long. That's it. The concentration is actually quite low. The concentration of DNA, the good samples that we get is about 1 nanogram per microliter. That's it. You don't get more than that. So you get highly fragmented DNA. You also get damaged DNA. So a lot of these C's get converted to T's. So because as DNA is, these samples are not stored. Okay, so most of my samples are now from the Lee Kuan Yew Natural History Museum in Singapore. Singapore, you need to understand, is a tropical country. It's hot and it's humid. It rains almost every other day. So these samples were not kept in air conditions. They were only shifted to air conditions about 30, 40 years back. So a lot of DNA actually gets damaged. So most of the people don't like to work from collections from museums in the tropics just because you will not get good quality DNA. And they are also prone to contamination from external sources. You'll have a lot of bacterial contamination and things like that. So there are a lot of challenges, but then these are the only samples you will get. And some of these museums have wonderful collections. So, you need to figure out different ways to actually try and probe them, try and get DNA from them and understand what were the patterns like. And what has really been helpful in the past 10 years is the application of next generation sequencing for non-model organisms and for ancient DNA work. We can now generate complete genomes for these samples. It's not a big deal. It doesn't take us more than a month to do that. So it's been a lot of work, but it has been a fun project to be a part of. This is what we set up. So this was the major thing that I did at Lee Kuan Yew at NUS when during my postdoc. We set up this ancient DNA facility. So we set up the whole room, designed the whole room, set up this facility, which is now being used by at least, I think, 20 different students who are doing multiple different ancient DNA projects. So now, general procedure for working with these samples are you extract your DNA in a dedicated facility. You do a whole genome library preparation. You can either sequence these whole genome libraries, or what we did is we used another step where we actually extract only small portions of the genome and then sequence them, and then look for again your genome-wide SNPs or sequence data. Now, this was the major step that we actually brought in. We designed our own probes and target regions from the genome. So we don't randomly sequence any portion of the genome. So we design our own loci. So what we did is, this is extremely useful when you work with birds. Their bird genomes are highly conserved. You look at all these 10,000 species and you can get most of the regions to align properly. So what we did is we actually looked at 3 different genomes. This was of a babbler. So this is a closely related species with the 2 species that I'm working on. We had done an in-house genome for this species as well. So what we did is took this genome and compared it to 2 different model bird genomes. So one is your zebra finch and the other is a collar flycatcher. So now collar flycatcher is also considered as a model for bird work genomics work. So you have a very good quality chromosome level assembly for both of these genomes. And ours is a non-chromosomal level assembly, but we can work with all of this. So basically, we aligned these different genomes, looked for exons which are at least 200 base pairs long, and these are single copy exons. So they are not available in multiple copies across the genome. So we've got these single copy exons. There should be at least 200 base pairs long. And what we did is we actually took sequences 500 base pair upstream and downstream of these exons. Now, the reason why we did that is we can then get intronic region. We are interested in looking at an interface of population genetics as well as phylogenetics. So we want information that is conserved enough to look at multiple species, but also variable enough that you can actually look at differences between populations. So this kind of design helped us do that. We could do phylogenetic work as well as population genetic work from the same data. So as soon as you get into introns, you get a lot of variable sites. So now once we've got, I think got about 10,000 such loci that we could take. We did a lot of filtering. So the major filtering that we did was we did a GC content filtering. So whenever we do NGS sequencing, it's better to have a tight range of GC content so that your sequences come out best. So we looked at 40 to 60% GC content. Then we removed any of these loci which had repeat regions. And after multiple such filtering steps, we had about 960 loci remaining, which could target about 2 MB of the genome. So these were the loci that we used further for our capture target enrichment. And these were sequenced across multiple samples now. So what were the main objectives of this study? So we wanted to actually understand colonization patterns of these islands in Southeast Asia. What about these small island populations? Are these admixed or are they unique? And what are the patterns of population divergence and speciation? So how does Pleistocene glaciation come and play a role in these in Sundaland? Now the two birds are here. That's your black-capped babbler and short-tailed babbler. Now what I'm showing you is a simple PCA plot as well as a discriminant function analysis plot. Now both of these actually just show you how the data is clustered. Now, in this, we are looking at only transversions because we are working with ancient DNA samples. So we had multiple data sets. I think we had about 12 different data sets. And I'm just showing you one of the data sets, how it looks like. So we are looking at transversions because these are rare. So these may not be due to any ancient DNA damage. So once we have your transversions, anywhere about 10,000 to 40,000 transversions were what we were looking at. What we see is we have these three major clusters. One is from Java, the other is from Borneo. So we have two populations of Borneo. One is Sarawak, one is Sabah. I'll just show you for those who are not aware of the geography. So this is your Sabah population and this is your Sarawak population. All right. So for the black-capped babbler, we have the Bornean island as forming one population cluster here. The Java population is separate. And in this, you have Peninsular Malaysia, Sumatra, Natuna, all of them clustering together. The same thing we see in a DAPC plot here. Here you have this major cluster which consists of Peninsular Malaysia, Sumatra, and Natuna. And this is your Borneo cluster, and this is your Java cluster. Now, short-tailed babbler is not found on Java islands. It's only found on Peninsular Malaysia, Sumatra, and Borneo, and these small islands. And what we see for short-tailed babbler is we have one really long spread of Sarawak. This is your Sabah population, very different, isolated. And then here you have the samples from Peninsular Malaysia, Natuna, Sumatra, and these small islands of Anambas and Natuna as well. So what we see from both these analyses is the Natuna island population actually goes and sticks with Peninsular Malaysia, not with Borneo as you would expect, because it's so close to Borneo, but it doesn't stick with them. The second thing is we actually can model gene flow patterns. We can actually look at how much gene flow has occurred between populations, when they diverge, what was the divergence like. And when you look at for black-capped babbler, you basically see is the first is your Borneo population separates out, then you have your Java population. So this separates out, then your Java. And then within this, what you have is Natuna, Sumatra, and Peninsular Malaysia. And you have a lot of integration, basically movement from populations that we have not sampled. And what we know is that whenever you had this Sundaland, this formation, you had populations here in Vietnam and all which we haven't sampled, which may have contributed. And currently these species are extinct from here. So we basically cannot sample them anymore. So basically these are gene flow integration from ghost unsampled populations. The other thing that we need to notice is there's this very long paleo channel here. So this may have restricted movement of birds from Natuna to Borneo. So even if they get connected, you have this huge, really uh, big river channel that flows that may prevent these small birds from flying across. The other bird, the short-tailed babbler, it has a more complex history. So you have your Borneo population also separates out. So you have Sabah, you have Sarawak, they separate out. And then the rest of them are closely related to Sarawak. And what we see is the island population of Anambas, Natuna separating out. And then you have Sumatra and Peninsular Malaysia. And again, we notice there's some movement from unsampled populations as well as from the ancestor into Sumatra. So there's a lot of complex movement of birds from different populations. There's a lot of gene flow. And what we see is always the Natuna is isolated from this Bornean complex. So what we actually need to think about is that just the location of the island is not just what drives the population's structure or the population composition. It's actually you need to look into the bathymetry. So you're basically looking at how the connections between islands were, and if there are any possible paleo channels or other areas that may prevent movement. And the Pleistocene glacial cycles may have played a major role here, because every time the landmass bridge were formed, you could have had movement between between Peninsular Malaysia and these small islands, Sumatra, but not between these. So basically your land bridges determine the genetic composition. It's not the distance to mainland, and complex gene flow patterns and population isolation patterns were observed. Now, paleo rivers may have played an important role, but right now we cannot test this. So we would need, if we had to go into this, we would need samples from all across Borneo here, and maybe even these smaller islands here to actually test out if these rivers played an important role. So till now, I've been talking to you about how Pleistocene glaciation may have played an important role in these island systems. I will shift to our montane habitat. If there are any questions, I could take them now or maybe even later, depending on how you want to talk about this. Because I'm going to now shift. Yes, please go ahead. Two of you have raised your hands.
So I was wondering how these trees were constructed, like maximum likelihood, maximum parsimony, what?
Okay. So yeah, just 1 second. I'll go back to this. So these are not your phylogenetic trees. These are known as admixture graphs. So these are actually constructed based on the level of gene flow. So have you guys come across something known as D statistics, Ababa analysis? I wasn't sure if you were doing this. So these are based on population drift as well as quantifying gene flow. These are not your phylogenetic trees.
All right, thanks.
Any other question? So see, the first clue for you that this is not a phylogenetic tree is you don't have a scale bar here. All right, this is just a pictorial representation. Yes, Ahmed, please go ahead. I think somebody raised a hand.
I just put my hand up, sorry.
Okay. Anybody else has a question?
No, I think that's it, Kritika. I can go.
All right. So we'll get into a different effect of Pleistocene glaciations now. So we are now looking at what would happen on a montane habitat. So just as a recap, so whenever you have the cooling phase, you have most of your water being locked up as ice. Your global temperatures decrease by roughly 7 degrees centigrade. And what we are now interested in is to look at how this would affect these montane birds. All right. So we are looking at the island of Papua New Guinea. So this is where we are. This is the area we are looking into. So we are looking at the highest peak there, that's called Mount Wilhelm. And it's about 4,500 meters high. This has a snow cap. Every time we have a cooling phase, the snow comes down to up to 3,500 meters. So a lot of species are then forced to go down the slope. All right, so this is the area we are looking at. So just imagine a nice mountain, and then you have species occupying the whole area. But as soon as you have a cooling phase, they have to come down just because most of the area gets covered by snow. Now we are looking at 3 different scrub wrens. These are small, tiny insectivorous birds. They all look alike. They're quite difficult to actually differentiate in field. Even experts have a tough time telling them apart. And they occupy this whole mountain range from somewhere around 1700 meters to up to 3700 meters. So just depending on different species, the buff scrub wren is found at the lower elevations. The large scrub wren is found throughout this elevation gradient, and the Papuan scrub wren is found on the mountain tops. Now, the objectives are to understand the effect of Pleistocene glaciation on both population structure and size, and how it interacts with species ecology. Now, again, here we are looking at single nucleotide polymorphisms. So you have your DNA, we are doing reduced representation libraries and getting thousands of SNPs. Now I'm showing you a network here. So here each sample is in an individual, and the color is based on the elevation at which it was captured. All right. So this is for the buff face scrub wren. You have 2 elevations at which it was captured, 1700 and 2200 meters. And basically what you see in this network is you don't see individuals separating out dramatically. The 2 clusters are connected to each other. That means there's no subdivision. Basically, if you capture the animal at 1700 meters, genomically, if you look at 2200 meters, they're the same. You look at in Papuan scrub wren also, across the different elevations, there's no subdivision. Same for the large scrub wren that actually occupies from about 2000 meters elevation. So basically, there's no segregation of populations across an elevational gradient. The next thing you want to do is then look at what was the impact of Pleistocene glaciation. So it's not making these populations differentiated. So we want to see what happens to the population size whenever you have a cooling phase. So whenever you have a cooling phase, the populations come down. So if they come down, they have a larger area to occupy, because when you come down the mountain, there's more area. And then we want to see if whenever they're coming down, do they see you see a range expansion for these species? Do you also see a population expansion or not? And you can model this. So you had 3 different models that we looked at. One is a constant population size. So there's no change in population size, whether it goes down the mountain or goes up the mountain. Wherever it comes down the mountain, there's an expansion, but we look at 2 different expansions. So you're looking at expansion during the cooling phase. So this is a model that looks at expansion in cooling phase. And this is a model that looks at expansion at the warming phase. So is it that when they're going up the mountain, they're having an expansion or not? So when you model these 3 and look at for all 3 species, we ran these models, and then we looked for the best model, did a model check, did a parameter estimation. What we see is all 3 species show an expansion roughly around 27,000 years back to 29,000 years back. So what is so unique about this period? So this is the time when actually your cooling periods was coming into a maximum. 20,000 years back, it was at its peak. So around this time, it started to get cold. And once it started to get cold, these populations had to come down. And once these populations come down, they have a largest area to occupy. So what we're seeing is all 3 species show an expansion at the same time. They were forced down the mountain during the cooling phase, and they show an expansion. Now, this is very interesting because if you look at cooling phases in the Northern Hemisphere, whenever you had a cooling phase, populations actually got restricted, and they show a signature of population shrinkage. So they actually show an a signal of bottleneck. So, it's interesting that, a lot of things last 10, 20 years back would be considered as, whenever you had the cooling phase, populations all contract and they all show a smaller range. They only when the when the climate becomes warmer, do they expand into different habitats and they show up population expansion. But the impact of Pleistocene glaciation depends on which area you're in. So in the tropics, in the mountain tops of tropics, during the cooling phase, you actually see an expansion, unlike what you would see in the Northern Hemisphere. So this is very interesting, so you have to actually do these studies in different regions. Yes, Deepa.
I was wondering if this model that you're using here is, I guess, agnostic to any competition or resource availability or any such thing during that period, right, in the happening. Do you have a sense for I mean, for example, if you do similar analyses of different populations of birds that would coexist in the same region, do you find that multiple birds would show the same kind of signature with the cooling? And then how would that impact, I guess, their ecological dynamics if they're all expanding into the same kind of area?
I think most of these birds would show an expansion pattern. So here we are looking at 3 different species of scrub wrens. They're showing an expansion. If we get into we have other birds also from there. We have samples from other birds. If we go into other warblers and all, we should see an expansion. I think most of them are going to show an expansion, but the timing of expansion might change.
Okay. And then what about, I guess what I'm wondering about is what happens to species that already existed in the low-lying areas, right?
So that's something that we have to get into. So if we have to then go into the base, we need to see what happens to them. That we don't know. So we are only looking at montane species. So we don't know what happens to species who are at the base.
Right.
Because what happens to their habitat? Is it shrinking, or are they having too much competition, so they actually reduce in size, or don't show a change at all? So we don't know that.
Right. Because you're also I'm also surprised at how much this expansion occurs, because I would expect the montane species to not be able to adapt so easily to the low-lying areas. But I mean, clearly they're doing it because you're seeing signatures of it, of this expansion in the. So I'm just thinking of what could allow that kind of expansion, because you wouldn't expect resource available to increase either, since it's a cooling period, and if anything, growth rates of everything should be a bit slower, perhaps, right?
I think it's just the range size increases, they get more habitat to occupy.
Okay, cool, thank you.
So basically what we see is we're seeing a population expansion during the cooling phase and homogenization of populations. So your major important take home notes are, sorry, Deepa, yes, go ahead.
Oh no, I forgot to lower my hand.
Okay, sorry. So the major take home points are basically the impact of Pleistocene glaciation cycle depends on the species biology as well as the habitat and location where they are found. So you can have contradicting effects depending on just where the species is located. Next, they have an important role to play in gene flow, population isolation, as well as colonization patterns. And finally, they have a strong impact on population demography. It can either lead to expansion or decline. Just want to acknowledge the different funding agencies. You had the Singapore Ministry of Education that, yes, Amir, I'll come to your questions. Let me just acknowledge people who have been involved in this. So I want to acknowledge the different funding agencies, the collaborators, as well as the museum collections, because without those, you can't do a lot of these studies. All right, now I'm ready to take questions. I can actually end the show. I can see people. Yeah. I think we can start off with Amir. Yes, please go ahead.
Sorry, I must have completely missed this, but how do you validate your model? And
yeah, so I did not get into all of those, Amir. So you actually have to do a comparison of different models. You look at the posterior probability of each model and then decide which is the best model. And once you actually find out that the model is, this is your model. So here we only looked at 3 models. So we got the model with expansion somewhere after 40,000 years back, right? After that, you can actually, we have done this where, we actually look at how much power our data has. So we actually generated different number of SNPs. We generated 500 SNPs, 1000 SNPs, 1500, 2000, 3000, 4000 SNPs. So you generate different number of SNP sets, data sets, and test the same models again there and see how many times will you actually get the same model. All right. After that, you also can do checks for, do you get the same pattern if you look at just 500 SNPs, or if you look at 1000 SNPs or look at 2000 SNPs. So you do a lot of different ways to validate your model. So you have to make pseudo data sets, run them, test them. And only if you get, in a huge proportion of your pseudo data sets, the same model, then you have some confidence on your model, right? So you have a posterior probability associated with each model.
Just to follow up on that, so you compare only these 3 models relatively or in absolute terms?
Only these 3 models. So for this, I've only compared these three models. I haven't made more complex models. We can make further complex models, like, you have an expansion followed by that. As soon as your warming period starts, is there a contraction? Then we are looking at only the past 100,000 years history, right? What about older than that? We are not making very complex models. We are making very simplistic models looking at one major event.
Okay, and you just measure the relative variation each model explains, right?
All right. So basically what you're going to do is you're going to make maybe 100 or 500 pseudo data sets. So you have your 2000 SNPs, right? So you're going to resample them, make 500 or 200,000 different pseudo data sets. Then use each data set and test which is the best model. For each of this, then you're going to get a probability, right? And then overall, you're going to get a probability how many times out of these 1,000 times when I tested with these pseudo data sets, model 2 was selected. That's your posterior probability of the model. And that's how you calculate the confidence in your model. Is that okay?
Yes, makes sense. Yeah, thank you.
Okay, any other questions?
Yeah, we went through a lot of different things about Pleistocene glaciation in Southeast Asia. So, there are people who are also working in India. They're looking at how, because India gets connected to Sri Lanka and Western Ghats at least. So they're looking at movements of birds within Western Ghats from India to Sri Lanka and vice versa, how much differentiation there is. Then with bats, I know there have been studies to look at how the climate in the past 10,000 years has impacted their population structure and things like that. So there's a lot of work that we can do in India also. It's just not been done because you have a huge peninsula, which actually becomes extremely arid. So it's not useful for a lot of species. They don't like that quality of habitat. Your Western Ghats is supposed to be a refugia during the cooling periods. Even Northeast is supposed to be a refugia. These are a lot of theories are there. You need to test them out. So there's a lot of things that we can actually do even in the Indian subcontinent with respect to these cycles.
Do the glaciation periods mean anything for the influx of species that would come into the Indian subcontinent? Apart from Sri Lanka, which seems obvious, but any of the other areas, they don't get affected, or that's not predicted?
Northeast should.
Okay.
You have a oriental inward migration you can have, because Northeast is supposed to be a refugia. So a lot of species would come, get stuck there, and then move out from there.
Makes sense. Yeah, I think in this particular school, we haven't had too much phylogenetic or other discussion so far. So this was good. People got introduced to it.
Yeah, I'm sorry I did not get into any of the details of the methods because it's just you won't go further away from one method if we can have a whole class on just one of these methods. So the idea was to get you to look at different things that we can do with the same data.
Makes sense. All right, if there are no further questions, let's thank Kritika once.
Thank you so much
for an interesting talk, and good luck setting up your new lab.
Yes, thank you so much.
See you.
Bye-bye.
Everybody else, we come back at 4:30 to listen to Guillaume's last lecture.
All right, bye-bye.